Good morning, everyone. Thank you for joining us today for the 10 collaboration webinars. We're really excited to have you here, and we're going to go over a few of the general information to get us started. So all attendees are going to be in listen-only mode throughout the webinar, and that's to provide better audio. We will also ask that if you would like to type in any questions that you have along the way or any comments, there's a question box, you can do that, and we may try and answer them. The presenters were saying that they may try and answer some along the way, but we'll definitely try and save some time to get to those at the end as well. We will try and unmute you also at the end to ask your question. So if you, hopefully that'll provide for better discussion. The presentation is going to be about 45 minutes or so, and then we're going to record it and have it posted on the Trial Innovation Network along with the presentation. And that'll be available tomorrow, and I'll show you where. It'll be under the archive tab under the events page. And excuse me. And if at the very end, if you'll take just a minute to fill out an exit survey, that would be great, because we always like to have your feedback. And if you want to volunteer to give a webinar, you can sign up as well. So I don't want to take any more time. I want to give the speakers as much time as possible. Today we have a team here that are going to be presenting using the National COVID Cohort Collaborative N3C data to inform your protocol development. And we have a group with us today. I'll introduce them briefly, and they'll introduce themselves a little bit more when they get started. We have Melissa Handel, we have Dan Hausman, we have Hamil Mehta, Carissa Madlock Brown, and Anita Waldron. I hope I didn't slaughter anybody's name. So I'm going to go ahead and pull up the slides and start at the beginning. And whenever you all are ready, I'm ready. So thank you for being here.
Terrific. Thank you so much, Bridget. So it's really, this is Melissa. It's really my pleasure to, you can go to the next slide, please. It's really my pleasure to introduce everyone here and to really kind of create a partnership, deepen our partnerships between TIN members and the CD2H, the CTSA Coordinating Center for Informatics. We've been hard at work on the National COVID Cohort Collaborative, and today's presentation is really aiming to support how can the N3C help support TIN study, design, and implementation, and deepen that partnership. So I'll go ahead and start off by talking about some of the work that we've been doing on the medical record data from across the CTSA consortium. Next slide, please. And then you can go to the next slide as well. So first of all, what kinds of questions can the National COVID Cohort Collaborative, or N3C, actually address? And so the N3C is a large-scale data integration effort across the CTSA and other medical centers. And we have really aimed to create this platform with a very large cohort of observational EHR data. And so we can ask questions such as, what social determinants of health are risk factors for mortality? Or do some therapies work better than others by region or by demographics? Can we compare local rare clinical observations with national occurrences? So really validating those findings in the face of this this novel disease at a scale where we can really understand if that's really happening everywhere. Can we predict who might have severe outcomes if they have COVID-19? What factors will predict the effectiveness of the vaccines? Can we predict acute kidney injury in COVID positive patients? Who might need a ventilator because of lung failure? Do some therapies work better than others? Next slide, please. So I wanted to speak about this through the lens of understanding long COVID. We know that we're going to be designing a number of studies relating to long COVID coming up. And so I thought this was a good example of how this incredible cohort of data is potentially useful to inform that. So for example, who has long COVID? Even just determining that is very challenging, and I'll get to that in a minute. But what we really want to understand is how effective are existing treatments, how do viral and host variants correlate with the outcomes, and how can we best design trials for long COVID patients? And then finally, how can we deploy, evaluate, and refine care guidelines quickly and effectively over time? So when we think about the longitudinality of COVID and frankly, any disease, we basically take data about a patient and we ship it off to different places. We have our EHR data, we have our genomic data, we have our imaging data, we have patient-reported data. And it's very hard to really get a complete picture of the longitudinal trajectory of a patient when these data streams are going off to different places. And so one of the things that we're doing in the N3C is trying to re-coordinate the alignment of these different data and help support disease classification. So for example, for understanding who has long COVID, the ICD code that's recently been created will not actually help us because we have yet to create a valid and reliable phenotype. Patients with the ICD code will be extremely heterogeneous and a poor cohort for designing prospective studies unless we do that disease classification. And so even with simply the electronic health record data available in the N3C, we can already start to stratify patients into different subgroups based upon different inclusion and exclusion criteria. And so in the long run, however, to define long COVID as well as any disease, we really need to be able to classify patients according to these multimodal data classifications and subclassifications, and that will ultimately result in the best study designs. Next slide, please. So I just wanted to give you a little bit of an overview about the characterization of the N3C cohort to date. This is a preprint of our characterization paper. It's at that link there at the bottom, bit.ly/cohort-preprint. And shown here are the WHO severity scales that we've aligned all the data from across the different sites. And you can see the overall percent and the different severity categories shown on the left. Over on the right, we can look at medication use, and here we've seen that severity has decreased over time, and the use of antimicrobials and immunomodulation drugs has shifted over time. So for example, you can see the decreasing systemic antibacterials are having a tendency to decrease in use over time, whereas we're seeing an increase in remdesivir and systemic steroids over time. Next slide. And so this is also in the preprint paper, and I should mention that this work was led by Dr. Tel Bennett at the University of Colorado, CTSA, and with a large team, of course, from across the network. And this is one of the outcomes of having such a large cohort of patients, was the ability to create a machine learning model from all of these different data. And in doing so, we basically, in this case, we took 32,000 COVID positive inpatients, adult patients in this case. There are pediatric patients in the cohort, but this was based on adult patients. And this machine learning model predicts mortality, IMV, or ECMO. And then provides a ranked list of predictive features. And so this just kind of gives you a sense of like the power of having this large data set where we can, from the first day of inpatient clinical values, can predict who is going to be severe in response and then manage their care based upon this in a highly confident, repeatable, clinically relevant, rapidly deployable, and easily refined method. And so our next task is to figure out how do we actually get this into the hands of the hospitals. And in this case, one of the other great things about the outcome is it's not only applicable to, sophisticated academic medical centers, but these are values that are readily obtained in any clinical setting. And so worldwide, such machine learning models can be very impactful. Next slide, please. So I just want to give you an overview of the timeline. Obviously, we've been hard at work since COVID first hit to try to figure out how can the CTSAs work together to be a true network and bring their data together for maximum impact. And so this timeline just shows our kickoff in April all the way to the present day, where we now have actually 78 data transfer agreements. So that's 78 institutions that have promised to share data. We have 39 sites now, today it's not current, that have the data harmonized from all those different sites. And this includes not only the CTSA communities, but also the IDEA Centers for Translational Research. And we also have OCHIN Rural Healthcare Network from across the nation also now contributing data. There are, so we're up at 2.8 million total patients in the cohort. And so that's a lot of patients to really think about, how to sort of validate your potential study designs against this large cohort for COVID studies. Next slide. So this just gives you a sense of the numbers. Again, we are roughly expecting around 3.6 million patients when we get those 78 sites all ingested, and we have that will mean over 8 billion total rows of data. And so it's really an unprecedented level of data, clinical data sharing across the nation, and we believe it's the largest limited data set available in US history. And I should also mention, and these numbers are now also out of date, but we have over 1,200 participants in the initiative and over 91 projects that are ongoing. So it's really rapidly become one of the largest team science and translational science to date as well. Next slide. So I want to introduce Dan Hausman. He's the Chief Technology Officer at Gradical, and he's going to talk to you about the work we've been doing to coordinate with the pharma community.
Hi, everybody. As Melissa said, I'm involved in real-world data. Let's see, did I get switched on? Can you go to the next slide? I just want to say that the purpose of the Pharma-Commercial domain team, and each domain team has its own area. We're a fairly different domain team in that we're really focused on external engagement to different groups in the commercial space. And for all of the amazing work that has been done to create this resource, there's still a lot of work to make sure that the groups that can most benefit patients, as well as their own organizations in using it, still has left to be done. And so we're focusing on reaching out to different groups. Can we go to the next slide? The groups we're most focused on are the ones that are involved in translating treatments into the patient communities. In the pharmaceutical space, we're working to look at groups across the spectrum, not just necessarily drugs that treat COVID-19, but also drugs that may have an effect in a COVID-19 population, drugs that may not have been considered because there could be a drug repurposing potential. We're looking at medical device companies who may provide tools such as ventilators or other measurement devices that could help COVID patients. We're interested in working with contract research organizations. And one of the groups involved closely in the N3C community is IQVIA, which has been supporting in conjunction with the Odyssey organization, which is an open source project for organizing data sets. And we also want to work with the consulting companies, you know, the large groups like Deloitte and PwC and the other groups that are helping the industry organizations to conduct their research, to have the kind of resources they need to use this data. And finally, we are also interested in engaging with the technology companies. You know, one of the organizations that's involved in some of the data management is Trinetics, and Trinetics also supports clinical trials. And so figuring out how we can work with these organizations to support their objectives is important to what our group's trying to do to get this information most effectively used. We go to the next slide. So what do we do as a group? We do outreach to these groups. We try to communicate the success from the groups that have already used the data and produced results. We're just now translating into the phase where we're starting to see publications that we put out. So we're excited that there should be 2 or 3 publications that were developed about 3 to 4 months ago that are going to finally be something we can communicate about. We do look to mitigate blockers. The organization, because it comes from a government and academic collaboration, wasn't always designed to support everything that commercial groups are looking for. And so, we're happy to look through if there's a gap. We've been working recently to look at ways we can communicate what's inside of the enclave more clearly, and collaborate with the other work groups to have a public-facing catalog that gets deeper into, for example, what drugs are represented and what populations are taking those drugs within the enclave data without having to go fully inside to do queries. And we work to figure out how to leverage this data to inform clinical trials. If you go to the next slide. In looking at the different use cases from industry, we're really focused, I think, when we look at clinical trial innovation around research speed. There's certainly other overlapping areas which may also come into play. For example, one of the groups that's working with the data is called ARI Biosciences, and they've been working on drug repurposing. So looking at the available known active pharmaceutical ingredients to see which target the specific biology of known preclinical research, and then trying to see if those drugs are having some effect. That's what's going on the drug repurposing side. Certainly groups like Novo Nordisk, who are very closely involved in diabetes and metabolic disorders, are looking at how do their drugs work within these populations, which involves sort of looking at the disease understanding. But from the research speed question, it's really about figuring out how we can make studies happen faster, because we can understand what's happening within the populations we're looking at, and to focus on solutions to recruiting the subpopulations that are of the most important categories that are going to supply the patients that'll prove out what that clinical trial will execute against. Early on, when we were looking at clinical trial type questions, the kinds of questions that were coming in were about what kind of study to build from a study design perspective around vaccinations. So this is before vaccines were out, and their questions were things like, what does a mild COVID case look like? What are asymptomatic patients going to look like? So they could model in a study how to know if a drug, in this case a vaccine, was effective or not. And so, we have this large data set that we think can very effectively help to model studies. If you go to the next slide. And, there's this broader potential, I think, if we can collaborate more deeply with sponsors or with CROs or the NIH funded projects as well, is to look at translation going both ways from the data sets into the clinical studies, as well as to the execution of the clinical studies. And so certainly the N3C enclave, the de-identified data store that comes from the health system, extracts and has a specific site extract that's focused on this narrow population. It's a very useful resource for doing the trial planning and for the modeling of what kind of logic to use to recruit patients. But within the sites themselves, there's also this very rich data source being regularly collected about these populations. And so there's the potential to use that data source by contracting directly with the sites. The N3C itself isn't really set up as a CRO to contract multiple sites, but it could enable use of that data, because that data is going to look like the same information in N3C enclave to help accelerate potentially using that information to support recruitment and engagement of patients in a study and working within the sites that are already part of the network. I think if we were able to execute this together, if you go to the next slide, there's some pretty good benefits in terms of both for the patients as well as for the groups that are interested in executing these studies. I mean, the big picture would be we can execute faster and better COVID-19 trials. But in terms of the capabilities of N3C, one, we'd have common tools in terms of the design as well as the execution, because we could use the same normalized data sets and extraction tools. I think we could really speed up the deployment and ramp up of trials by sharing some of the infrastructure. The data normalization could really streamline some of the complexities that normally come into the recruitment process. I think we shouldn't overlook the huge benefit of the expertise, both about the data as well as about COVID-19 within the N3C community. I think people get focused on the volume of the data, but it's just as important to look at the people who can become principal investigators at different sites, and their clinicians that are regularly researching various different categories of disease. And there's the skill sets within the community to execute pieces of data science and technology that usually don't exist when you're just working with an individual site to deploy a research study. And then finally, I think the The sites that are already participating can benefit from some additional sponsored funding to build out the investments they're making into the data, but also to support their patient communities and their researchers, and having the appropriate governance that can leverage some of the existing governance, which really wasn't set up for executing interventional studies, can be used to accelerate trial setup and to organize the groups that are already involved. So, I think there's a lot of opportunity of faster, better COVID-19 trials. As a summary, the system wasn't designed today to be a clinical trial recruitment engine. It's really an enclave for doing retrospective research, but it is possible to leverage what's already been built to really help out groups trying to do clinical trial execution. So thanks.
Hello, my name is Hemil Mehta. I'm a pharmacist by training and assistant professor at Johns Hopkins Bloomberg School of Public Health. My research mainly focuses on pharmacoepidemiology. Next slide, please. So the main difference between clinical trial and what we do in pharmacoepi is we rely on real world data. It could be like healthcare claims or the electronic medical records data to look at drug use, safety, and effectiveness. And the difference in pharmacoepi is treatments are not assigned randomly as it's done in clinical trials. So that poses a challenge. How do we control for confounding and selection bias, which is implicitly controlled when you randomize people? And that also opens up a new area of research, which is methods, like how do we control confounding and bias while evaluating safety and effectiveness of treatments. We use large database, which includes large number of people, for example, N3C. And the beauty of this type of research is results are generalizable to real world population. So in randomized trials, they have the advantage of internal validity versus pharmacoepidemiological research. They have the advantage of external validity, which is generalizability. Next slide, please. So in N3C, our mission is to generate fundamental new knowledge regarding how drugs are used, how safe and effective they are for the treatment of COVID-19 infection. And for this, we rely on this large N3C data to do high quality observational research on drugs, for example, remdesivir, dexamethasone, oral anticoagulants. We also have projects going on anti-cancer drugs, drug-drug interactions, and all these projects will generate new knowledge, which can help clinicians and policymakers. And the outcome, we want to make new knowledge and have the clinical value for COVID-19 treatment, which can help stakeholders. Next slide, please. So as in this slide, we just show some examples of active projects. So we have more than 10 ongoing projects. So first, for utilization, we are doing one study where we are looking at how hydroxychloroquine, remdesivir, and dexamethasones are used. The other projects focuses on oral anticoagulants. So trends in use over time, how drug use differs across centers, how drug use differs across patient characteristics. The second set of projects focuses on safety of these drugs in patients with COVID-19. So for example, one project focuses on thromboembolic events associated with drug-drug interactions, in this case, specifically dexamethasone and oral anticoagulants. We are also looking at how remdesivir, dexamethasone, and anticoagulants are used, and what are their effectiveness on clinical outcomes in patients with COVID. And as I said earlier, patients are not randomized in real world practice. They receive drug A versus drug B based on certain clinical characteristics. So we need to account for these things. So the other type of research, we do methods research. So in this case, one example is some analytical challenges, for example, healthy user effect. So we are doing that type of research also. Next slide, please. So just to give you a sneak peek of one project we are doing on drug utilization. So here on x-axis, you can see time from March to October. And on y-axis, we have % of individuals, hospitalized individuals receiving products. The green line shows hydroxychloroquine. So you can see that in March, April, the use of hydroxychloroquine reached to approximately 40%. And then starting early May, it just went down. And currently it's between 1 and 2%. And we have in boxes, you can see that the emergency use authorization was done on March 20, and then FDA revoked. They issued caution, and then ultimately they revoked EUA. So with respect to these major events, how the drug use has changed over time. The purple box is for remdesivir, and you can see that starting from May, the use has increased. Still, we see that approximately 17 to 18% of patients receive remdesivir by the end of October. And the orange line is for dexamethasone. You can see that after preliminary results of recovery trial came in mid-June, the use really spiked up. And by the end of October, the use reached to approximately 40%. And we have done some additional analysis looking at people who are ventilated versus those who are not ventilated, how drug use differs, what are patient characteristics associated with drug use, like the socio-demographic or clinical characteristics, and also how drug use varies across different centers. And we found some really interesting findings. We are in the process of writing this paper, and hopefully we will submit this week. Next slide, please. So here is another research question, grad student from Hopkins working on. She's a part of N3C community. And her question is, do individuals who are on chronic immunosuppressive medications have worse outcomes compared to those who are not? So in this case, real world evidence of pharmacoepidemiologic studies using N3C data are really useful because it may be difficult to do clinical trials for this type of questions, and you may not have enough sample size. And also it's time consuming and expensive. So for this, we are using N3C data, and we are using propensity score analysis to control for selection bias and confounding. And the clinical outcomes we are evaluating are mechanical ventilation, length of hospital stay, and 30-day mortality. So we did this study with local data from Hopkins, COVID-19 registry data, and we found that chronic use of immunosuppressive medication was neither associated with worse or better clinical outcome among patients with COVID-19. Next slide, please. So that project was published in Clinical Infectious Disease Journal. So here we just show the snapshot of that article. And also second article from our team, Caleb Alexander, he is a co-lead for Pharmacoepi clinical domain team. He also published a viewpoint in JAMA. Next slide. So yeah, we have over 30 members in Pharmacoepidemiology domain team from government organizations, universities, and it's a fertile ground for collaborations. We have over 10 ongoing projects. We meet every Monday, every other Monday from 4 to 5 p.m. Eastern Standard Time. We have actually a meeting today, so if any of you are interested, please shoot me an email. And there are opportunities for leadership, for current or new investigations. As we know, every week you learn something about drug this is effective or this is not effective based on ongoing clinical trials or based on clinical observations. And N3C provides data, and we have research expertise to do some of these projects using this data and generate real world evidence. Thank you. Next slide. Charisse.
Yes, hello. My name is Charisse Madlock Brown. I'm an assistant professor of health informatics and information management from the University of Tennessee, and I'm the domain team lead for social determinants of health. Next slide, please. So just to give you a sense of what we're talking about here, these are social determinants of health are the circumstances in which people are born, grow up, live, work, and age, and the systems put in place to deal with illness. These circumstances are in turn shaped by a wider set of forces. So really the reason we are interested in looking at social determinants of health in COVID-19 is that there are all sorts of variables that are going to impact health that don't really have anything to do with pre-existing conditions necessarily or different healthcare related variables. So for instance, people's access to health and how likely and often they are to even seek care. At what point do they actually get to the hospital? Are there certain groups where they're waiting till things are severe and they go to the hospital, or there are people doing preventive, doing more preventive care? So it could be around things like behaviors. The other issue is that people could be in very different types of environments. So for instance, we know that there are some areas where people are better able to social distance than others. These could be because of how densely populated the areas are. We know that house size can make it challenging. You know, how many people are you've got a lot of people packed in like apartments, and you've got a lot of people in those apartments. That can complicate matters. And there are issues around who's working in essential services or not. And then of course, there are just other social determinants of health, not specific to COVID-19, things like air quality, poverty, lots of different issues can impact your healthcare outcomes. And so our group are looking at some of these factors. Next slide, please. So we have quite a few activities because social determinants of health is very broad. That's one of the things that makes our group very unique, is that we're able to look at there's so many different variables we can look at, and so many different types of issues you can have between social determinants of health and COVID-19. So for instance, one of the things we're looking at as a social determinant of health is the local policy around COVID-19. So we've actually identified several different COVID-19 specific social determinants of health that we're looking at. So it could be, when did a given community first have shelter in place with respect to when did they have the first case within that community? So, were there many cases before there was any policy, something like that. Another sort of COVID related issue could be around actual shelter in place behavior. So we've got some data where we can look at how often were people actually moving around, or were there times where people really were sheltering in place. So those are some of the things that we're looking at. We also want to look at the impact on disadvantaged groups. So there's already been plenty of research out there showing that there are certain groups that are more affected. Maybe they have higher cases, maybe they have more severity. Now, some of those groups are historically disadvantaged minority groups, and some of them aren't necessarily. So we know that despite, while Asian Americans typically aren't as disadvantaged as African Americans, they're also being more affected by COVID. And then the other thing we're interested in looking at is how the impact of the pandemic on inequality. There could be areas where unemployment is increasing and different things like that because of the pandemic. Certain businesses, there could be areas where businesses are really floundering. So we're interested in looking at how the pandemic has maybe exacerbated or increased these inequalities. There's also been issues around seeking care, particularly in the first few months of the pandemic, where fewer people were showing up at ERs for the kinds of ailments they would typically show up. So it's suggesting that a lot of people were avoiding seeking care for other conditions. Now, another thing that is kind of a big initiative for us is actually looking at our research data, because a lot of these social determinants of health data are not already in the enclave. So we're actually looking at the limited data set. So there's different levels of access within the enclave, and we are using, we're getting the level of access where we can look at people's zip codes so that we can look at the area that they're in, and be able to look at different areas and understand what might be going on with, for instance, air quality in that area, level of poverty, density, access to healthcare, food insecurity issues, all that sorts of things. So we're getting a lot of other data sets into the enclave, and we're mapping those to, we're using zip codes to map those, and we're mapping data that might be outside public data that might be at the county or zip code level. And then anybody who kind of joins our team can really benefit from the richness of our data. So we're also looking at, we're able to get some data on the essential workforce. We have monthly unemployment rates. We have shelter in place behavior, which I talked about before. So we're able to, for a given area, look at how often on average people might be be away from home, for instance, and that's based on people's cell phone usage. There are a lot of already social determinant of health indicators at the county and sometimes at the zip code level. So there's something called like a social deprivation index. So what we're doing is we're still working on building out a data set that links to the N3C so that we can very richly do this type of analysis. The other, one of our other efforts around research data is we really want to improve the social determinant of health data that's in the enclave. There are many of these sites that might be using surveys around social determinants of health. So they might be asking questions around homelessness, food insecurity, and other social determinants of health. But they might not be currently pushing that data up in the enclave. And so we're working on trying to make it easier for people to push that data, no matter how it's represented in their local system. So that's one of our big initiatives, is to actually improve the social determinants of health quality. Then we do a lot of hackathons on not just training the system, but quality control. So since we're using so many different data, we really need to make sure that we don't have quality issues. Next slide. So while we're working on quite a few projects related to social determinants of health, and most of those are around looking at which social determinants of health factors are associated with either COVID-19 severity or are associated just with high incidence, maybe at the county or zip code level. We're also looking at differences by race and ethnicity. But I did want to just talk about why social determinants of health are just really important for all kinds of studies outside what we're doing. So one of the issues is that research shows social determinants of health are associated with poor health outcomes. And this is including for COVID-19. So this could be an unmeasured confounder in many studies. It could potentially be helpful, for instance, if you're doing risk stratification, to include some social determinants of health elements. Social determinants of health can potentially sway clinical trials, as patients with those characteristics often have worse outcomes regardless of treatment plans. So there's been research recently into this, and this is what they found. So it can be very important to incorporate these elements. And that research has shown that social determinants of health are associated with poor study retention, independent of age, gender, and race. So really, so this could really impact the planning around clinical trials. There's also several screening tools that exist to help researchers account for social determinants of health. So actually, when I said that we are wanting to promote the capturing and mapping to our system of social determinants of health, I'm talking about some of these validated screening tools that are typically used. So these are some of the screening tools we would like to see, more people using and getting that data pushed to the system. And these include the Accountable Healthcare Health Communities Health-Related Social Needs Screening Tool, something called Prepare, and Clear. And I just have a list of some of the citations for the claims, but social determinants of health We've got a group where we've got some of our specific instance or research questions that we're looking at, but we're also very open to helping other people because there can be a lot of potential for cross connect with our group and the other domain teams. Next slide.
Hello, everyone. My name is Anita Walden. I am the Assistant Director for Center for Data Health here at Oregon Health and Science University. So you just heard from a few other domain teams. On our website, we do have a list of probably about 20 domain teams in areas such as pregnancy, acute kidney injury, and imaging. I just put into chat the list, the link to the email, to the webpage, so you can take a look at some of those domain teams. Next slide. So these domain teams are multidisciplinary. As you heard, there are people who are clinicians who are on these teams, statisticians, informaticists, people from pharma companies or from the community. So they're all working together in order to solve the COVID, answer COVID questions and solve, this crisis. But this is also, there are other benefits to being part of the domain team. And that is, this is an opportunity to have access to one of the largest COVID data sets currently right now with EHR data. If you're a group who doesn't always have access to EHR data, you can get access here, and I'll tell you about how to do that in just a moment. Also, you can also use this data set in order to get some pilot data for research grants. So it's great for your TL1 and KL2 scholars, and also if you have any other grad students, or we even have high school students who are working with us for projects as well. And so next slide. Oh, before I actually go back one moment. So one thing I just wanted to mention is if you want to learn a little bit more what it's like to work on the domain team, up in the right-hand corner here, I just have a screenshot of Dr. David Ellison. He's a clinician. He's director of the Oregon Clinical Translational Research Institute. He's a member of the AKI domain team, and he gave a brief description of his experience. So there's a clip on the domain team website. If you're interested in learning a little bit more, you just click on that. It's really short and brief, and you can also feel free to share it internally. Next slide. All right, so how to get involved. And so in order to access the enclave, you do need a data use agreement. And also I place in chat a list of those organizations who already have one. So your organization may have a data use agreement in place. So check the website and see right now if you actually have one or not. If you don't have a data use agreement in place, your organization does it, you can get one in place. You just work with the National Institute of Health and CATS, and they'll work with you to get a data use agreement in place. And so once you have a data use agreement in place, you just need to sign up for an account, and then you can get access to the enclave. There is a data use request form you also need to complete, but it's electronic and it's within the system. But in order to know exactly the steps you need to follow, we have an enclave checklist. So look at that checklist. And follow that checklist. So there are different ways to engage with a project. So you can either create your own project if you have a clinical trial that you're interested in getting some data for, or pilot data for. You can actually create a DUR yourself, a data use request yourself to do that. Or there are existing projects that you can join. As you just heard from these three, they're working on different projects. You can enjoy it and you can collaborate with them on those projects. And there's ways to do that. So there's different ways to engage. We also have training. So if, there's a lot of information we're throwing at you right now at one time, but we do have training. So their office hours are located, office hours are Tuesdays and Thursdays. The information is also located on our website, is at 2 p.m. Eastern time. And we actually have an orientation come up, which I'll go through in just a moment. And there are tutorials available. So if you're new to N3C, or if you're not new and you just want to learn how to use some of the different tools, there are tutorials available for that as well. Next slide. All right, so here's information about the orientation session. So a lot of people said, well, I'm brand new to N3C. I want to learn a little bit more. Actually, starting tomorrow, there's on February 2nd at 11 a.m. Eastern time, there's a part A tutorial. Pretty much is like an overview of N3C. tells you actually how to get your account so you can log on and access to the enclave. And then there's a part B that's going to be held on February 9th. Once you actually submit a data use request, that means you have your own project that you're working on inside the enclave, and you want to know actually how to navigate within the enclave. That's what that session is for. And so there's a recommendation that you have session A before session B. But if you already have a data use, if you already have a data use request, that means you already have a project that's been requested in the enclave, then you can just do session B. And then we'll hold these sessions on a regular routine schedule. This is our first kickoff for the very first sessions. So please continue to check the website to see when there are additional sessions coming up. Next slide. All right, so we're encouraging you guys to get involved. We're encouraging you to do your own research projects. We're encouraging you to use this, wonderful database, for your clinical trials and research opportunities. Here's a funding opportunity. So if you need funding, there's a brand new opportunity. It's an R03, it's a small grant program. And so this is, if you're looking for a little bit of, funding, check this out. And here are some examples here of what you can use that funding for. But if you're still, not quite sure, NCATS has said, please talk to them. So if you have a project that you're interested in doing, they would like for you to talk to them on guidance on how and which type of funding to apply for. So definitely have those conversations with NIH about funding and or support your projects, especially if you're going to engage with the domain teams. All right, next slide. All right, so kind of to summarize, as far as next steps, check out the resources we discussed today and look at the domain team website. If you haven't registered for N3C, please register. Registration will allow you to get access to our documents. Everything is open. It's just that we need to, make sure everybody's registered. To access the documents, to get signed up for any meetings you want to participate in. Visit the N3 website, especially the domain team page. And then, once you actually create your account, take time to just browse around in the enclave, see what's there, look at the different training resources. As Cherise mentioned, that we did bring in some external data sets, so those would be there as well. And if you have questions, all of us here are here to help. I mean, one of the things I can say is that this has been a fantastic group. It's been a network of people from all across the country working together on trying to solve this problem. So just reach out to us or anybody else who who's part of N3C to help answer questions. And then we also have a support desk. So, you can always email the support desk with any of your questions. We review that information ongoing. Next slide. As Melissa mentioned earlier, we do have some publications that are out. This is the preprint that we just mentioned earlier. Next slide. And we also have a methodology paper that came out earlier, and we had about 100 authors. So one of the things we strongly advise and strongly support is collaborative research and people getting credit for the work that they're doing. So definitely, if you're interested in in publications, this is a group to participate in. Next slide. All right, there's some communication materials that Andrea Volz, our communication manager, has put together. So if you want to communicate information internally to your groups, and we encourage you to do that, please check out the website there. And there's NCATS and both N3C put together a toolkit, media toolkit for you. Next slide. And also, if you're interested in N3C in general, as far as the operations of it, we have different work groups, from governance to synthetic data. One of the things we didn't mention here, there is a synthetic data. We do have a synthetic data that's available too. And then and collaborative analytics work stream. So you're welcome to participate in the operations. And I placed a link here just to remind you if you want to join, and join means just, get involved or, just kind of stay tuned. You can register, and we send out information about the latest things that are happening. Next slide. And I just want to say this is a great resource, especially for cohort identification. It can help you with trial designs. And we hope you'll take advantage of this rich resource, not just of the data, but also the community that's come together. It's a great, there are a lot of experts around. So it's a great opportunity to get access to some rich EHR data. And so we're just going to pause here to see if there are any questions.
Thank you guys so much for that wonderful presentation. We have a couple of questions in the chat box about the slides being available. We will post the slides from today and the recording on the Trial Innovation Network. And at the end of the call today, Bridget will show you that and pull it up the events archive page and show it to you where you can find them tomorrow. But as of right now, those are the only questions I see in the chat box. If anybody has any questions, you can raise your hand and we can unmute you, or you can type it into the question and chat box. Oh, I do see a question from Charlie. Let me try to unmute you. There you go, Charlie, you're unmuted if you'd like
Hi,
to.
everyone, my name is Charlie. Thanks so much for the presentation. I was just curious, I guess in the short term or the long term, whether or not you saw work with EHR and databases, maybe per institution or across institution, replacing or somewhat replacing control arms in clinical trials.
That's a very good question, Charlie. Thank you for that. You know, I think it's, and I'd love to hear others' thoughts on this as well, but I think the answer is sort of, because the reality is that what we can do is we can actually really, I hope over time with this, you know, really what is an experiment in terms of, you know, having this access to this very, very large cohort with controls in the context of the COVID, that we can actually design much more robust trials based upon, you know, sort of quote controls in that context. It may mean that control arms are, I don't think control arms are going to go away, but it may mean that they could either be smaller or more narrowly focused based upon the findings from these large cohorts. And I guess this has never been done before, so I don't know the answer, but I'd love to hear anyone's thoughts about you know, what access to this kind of large scale data would mean for designing the control arms.
Well, this is Dan. I mean, at least in other areas, not necessarily in COVID, you know, we are seeing some control arms being put together specifically in diseases where it's, you know, a fatal disease with a very narrow window to treat, where it's potentially unethical to throw a control arm into a very high likelihood solution. But those are a fairly narrow scenario. And it, you know, I think the idea, much to what you heard from Melissa, is sort of is going to be the right answer, because an RCT has a very specific aim to control a lot of the variables during the treatment that we'll never be able to do within observational data retrospectively. But, you know, there will be a role for it, and it'll, you know, enhance the intelligence and reduce the scale needed to get the control arms put together. So I think we'll just see a lot of odd combinations of EHR data supporting pieces of the control arm, and maybe reduce some of the use of placebos or use of alternative arms. And we're also seeing now sort of these basket study type designs where there's multiple different interventions all being tested against the same control. And it'll be interesting to see how that intersects with initiatives like N3C.
Great, thank you for that answer. We have another question from Zoe. Zoe, I've just unmuted you if you'd like to ask your question.
Hi, everyone. Thank you for a great presentation. My question is really around the implications for recruitment. So I understand how this large data set may you know, inform the feasibility of conducting a study, as well as, of course, the design and the inclusion criteria. But have you guys been able to see the impact it may have on how teams are selecting the recruitment methods that they're using at all, and the kind of circling back and closing the loop with successes there? Just curious if you've seen anything to comment on in that area.
So I can start, but again, I would love to hear others' thoughts as well. I think, you know, it's really only fairly recently that the cohort characterization has been completed, and we have enough data to sort of do the kind of analytics and have the data harmonized. So we're you know, unfortunately, because our nation's healthcare data is so fragmented, and non-interoperable, it's been quite the long lift to even get us to this point. So we haven't really actually had the opportunity to do that. And I think that's one of the main reasons why we're here today, is to actually discuss that type of question where, this is unconsented data, and it's de-identified to the point that we didn't really talk about this today, but we essentially have dates of service and geocodes, but the actual information about the patients is not being itself submitted to N3C. So N3C cannot, and N3C is not allowed to contact the patients. However, what we could do is provide reports back to sites about their own sites, sort of contextualized cohort in the broader context of the nation, that could then help them thereby select inclusion and exclusion criteria based upon their own institutional attributes. And I think that might be a good way of supporting recruitment and candidacy for studies without not following our regulatory requirements. But I also think this kind of begs a broader question as well. So one of the big challenges, and one of the reasons I put the long COVID information in there, is that we're really challenged in the context of patient-reported information, and really to get a complete picture of COVID, and I think for many other diseases as well, actually really need that information to come in as well. And how should we get that information? And should it be via the sites, in which case there could be some portion that's consented? And so these are some of the open questions that we're somewhat battling with, and would really love partnership with you all to try to figure out, to help define what requirements are there that we could help serve up to you to help you with your trial recruitment and design.
Thank you so much.
Yeah, I think I'd say we're just coming into that. I mean, there's some folks who have just contacted me within the Pharma-Commercial group around, how can we leverage N3C to figure out how to recruit for a study that's already been funded. And, we're going to work through some of those in the next few months, because just now are some of these studies in a position to think about how to use the data, the infrastructure, the partnerships to recruit into their studies. So it's a great time to pose some real recruitment challenges and see what can be done within the infrastructure.
Absolutely. Thank you.
Well, I want to thank everyone for joining us today, and especially the presenters who pulled together a wonderful fact-filled presentation. And I want to show everyone where this will be. This is the trialinnovationnetwork.org website, and we will be posting this, and it'll be available tomorrow under the archive tab. You'll just click there, and then you'll be able to download the slides as well as the recording. We have a few more webinars coming up next month and the following months, and so be sure to check those out while you're there in case you want to register to listen in. And if you'd like to present and share your team's work and best practices and lessons learned, this is where you do that. So I really appreciate your being here today, all of you. It was a great presentation, lots of great information. And I hope that you, some folks decide that this is where they'd like to collaborate as well and help drive this forward, because it sounds like it's a very valuable data set that you've established and shared. So that's great. All right. Thanks. Yeah. Thanks, everyone.
Thanks, everybody.
Bye, everyone.
Bye.
Thank you. Bye.